Today in this class, we will discuss about the male external genitalia. The male external genitalia is the testis. It is the male gonad homologous with the ovary in females. It lies suspended in the scrotum by spermatic cord. The scrotum is a cutaneous bag containing right and left testis, epididymis, and lower part of the spermatic cord. The scrotum is made up from outside inward skin, dartus muscle, external spermatic fascia, cremastic muscle and fascia, and internal spermatic fascia. This testis is covered from outside inward by tunica vaginalis, tunica albuginea, and tunica vasculosa. Scrotum is divided externally into right and left parts by a ridge or raphe, continued on the undersurface of the penis and backwards along the middle of the perineum to the anus. Here in this section, you can see the scrotum. This is a skin bag of skin. Here you can see the skin, and deep to this is the dartus muscle, which replaces the superficial fascia. And here you can see it sends a septum, that is the median septum, divides the scrotal sac into a right and a left half. Each half contains the testis and epididymis and vas deferens. This testis and epididymis, you can see, they are enclosed in a bag of peritoneum, that is the tunica vaginalis. And here you can see the two layers, the parietal layer of tunica vaginalis and the visceral layer here. Here you can see the visceral layer adhered on the tunica albuginea, adhered on the tunica albuginea, that is a fibrous layer made up of fibrous layer covering the testis, and this is the testis. Here what you can see at the posterior border of the testis, you can see the epididymis. Here is the epididymis, and here you can see the vas deferens. Here deep to the dartus muscle, you can see here the external spermatic fascia. Here you can see the external spermatic fascia. This dartus muscle is replaces the superficial fascia of the scrotum and is prolonged into the median vertical septum between the two halves of the scrotum. It regulates the temperature within the scrotum. The external spermatic fascia, it is derived from the external oblique muscle of the abdomen. The cremastic muscle and fascia is derived from the internal oblique muscle. Its contraction elevates the testis, and this cremastic muscle can be stimulated by tapping on the medial side of the thigh, and this elevation of the testis is known as cremastic reflex. So, tapping the medial upper part of the medial side of the thigh stimulates the L1 segment, that is the ilium nerve, and this causes a contraction of the uh, cremastic muscle. This elevates the testis, and this is known as cremastic reflex. The internal spermatic fascia is derived from fascia transversalis. The tunica vaginalis is derived from the processus vaginalis. This processus vaginalis is a peritoneal pouch in front of the testis. It is carried by the testis during its descent from the abdomen. This tunica albuginea is, is the outer, dense, white fibrous coat of the testis. Then coming to the spermatic card. The spermatic card is a card-like structure in the males. It is formed by the vas deferens or the ductus deferens, and it is surrounding tissue that runs from the deep inguinal ring down to the each testicle. The constituents of the spermatic cord are the ductus deferens, the testicular and cremastic arteries and artery of the ductus deferens, then the pampliform plexus of veins, then the lymph vessels from testis, then genital branch of the genitofemoral nerve, sympathetic nerve plexus around the artery of the ductus deferens, and the visceral nerve fibers. Then the remains of the processus vaginalis. Coverings of the spermatic cord from within outwards are the internal spermatic fascia. It is derived from fascia transversalis. It covers the whole extent of the spermatic cord. The cremastic fascia. It is made up of muscle loops forming cremastic muscle and intervening alveolar tissue. It is derived from internal oblique and transversus abdominis. Hence, it covers the spermatic cord below the level of these muscles. Then the external spermatic fascia. This is derived from the external oblique aponeurosis. It covers the cord below the superficial inguinal ring. Here, this dissection shows the uh, spermatic cord and its covering of the testis. Here you can see the external spermatic fascia covering the testis, and the spermatic cord is the cremastic fascia, and the innermost is the internal spermatic fascia. And here you can see the structures present in the spermatic cord. That is the you can see the vas deferens, then testicular artery, then the artery of the vas deferens you find, and the pampliform plexus of veins. Testis is a male gonad suspended in the scrotum by spermatic cord. It lies obliquely so that the upper pole is tilted forwards and laterally. The left testis is slightly lower than the right one. The testis is oval in shape. It is compressed from side to side, 3.575 centimeter long, 2.5 centimeter broad from before backwards, and 1.8 centimeter thick from side to side, and weighs about 10 grams in adults. The external features of the testis consist of two poles, that is the apparent pole, lower. There are two borders, that is the anterior and posterior, and it has two surfaces, that is medial and lateral. The upper and lower poles are convex and smooth. The upper pole provides attachment to the head of the epidermis. The anterior border is convex and smooth. It is fully covered by tunica vaginalis. The posterior border is straight, it only partly covered by tunica vaginalis. Epididymis lies along the lateral part of the posterior border. The medial surface of the epididymis is separated from the testis by an extension of the cavity of the tunica vaginalis, that is the sinus of the epididymis. The medial and lateral surfaces are convex and smooth. Attached to the upper border of the testis, there is a small oval body that is known as appendix of the testis. This appendix of the testis is a remnant of the paramesonephric duct. Here, in this dissection, you can see the testis, and at the upper pole of the testis, here you can see the appendix of the testis. This is the appendix of the testis, this one. Then this is the head of the epididymis, this is the fat, and you can see the testis and the epididymis, they are covered by the tunica vaginalis. And here you can see the parietal layer, and here is the visceral layer is covering the, is fused with the tunica albuginea. Here, what you can see, the spermatic cord having the vas deferens, the testicular artery, and the pemptiform plexus of veins. The structure of the testis. Now, this glandular part contains 200 to 300 tubules. So, the glandular part of the testis contains 200 to 300 tubules. Each lobules Each lobule contains 2 to 3 highly coiled seminiferous tubules. Each tubule measures about 60 centimeters in length and 0.2 millimeter in diameter. It is lined by stages of cells forming spermatozoa, and this process of formation of the spermatozoa is known as spermatogenesis. The seminiferous tubules, they join together at the apexes of the globules to form 20 to 30 straight tubules which enter the mediastinum. Here, they anastomose with each other to form a network of tubules known as retitestis. The retitestis gives rise to 12 to 30 efferent ductules which emerge at the upper pole of the testis to enter into the epididymis. Here, each tubule becomes highly coiled to form lobe of the head of the epididymis. Tubules end in a single duct which is coiled on itself to form body and tail of the epididymis, and this duct is further continued as a ductus deferens. Here you can see in this diagram the structure of the testis. This is the outermost layer, that is the tunica albuginea, made up of fibrous tissue. Its end septa, that is the, I am dividing the substance of the testis into lobules, and each lobule contains some 2 to 3 seminiferous tubules. Here what you can see is the retitestis and the posterior border, and this at the upper pole of the testis, this retitestis is continuous with the head of the epididymis by efferent ductus. So these are the efferent ductus. This is the head of the epididymis, and here you can see middle part is the body and lower part is the tail of the epididymis, and this is continuous here you can see with the ductus difference or the vas difference. Here in this dissection you can see the testis from the lateral aspect, and here attached on the posterior border you find the epididymis. Here this is the this enlarged part at the upper end is the head, this is the middle part that is the body, and this is the lower part that is the tail of the epididymis. And this tail is continuous with the ductus difference and the vas difference. Here you can see the head, it is connected to the upper pole by efferent ducts. This vas difference here you can see it is accompanied by the artery of the vas difference, and this is what you can see is the testicular artery. The arterial supply of the testis is derived from the testicular artery, which is a branch of the abdominal aorta given at the level of L2. This testicular artery descends on the posterior abdominal wall to deep inguinal ring where it enters the spermatic car. The, at the posterior border of the testis, this testicular artery divides into branches. The branches are of two types, the smaller and larger. Smaller branches enter the posterior border, while the larger branches they are divided into two groups, that is medial and lateral. And these medial and lateral branches they pierce the tunica albuginea, they run on the surface of the testis to ramify into tunica vasculosa. Here, here you can see the testicular vessels in the abdomen. Here what you can see is the abdominal aorta, which divides here into the common iliac arteries, and here this is giving the branch that is the testicular artery. The abdominal aorta gives the branch that is the testicular artery which supplies the testis. While the testicular vein, here you can see on the right side drains into the inferior vena cava here. This is what you can see is the inferior vein. While on the left side, it drains into the left renal vein. This is what you can see is the tendon of the psoas minor. Here, the testis and epididymis has been viewed from the posterior aspect. Here you can see the head of the epididymis, this is the body of the epididymis, and this is the tail, which is continuous with the vas difference. And this is what you can see is the testicular artery. Venous drainage. The veins emerging from the testis form pampiform plexus. This pampiform plexus is divided into three parts. The anterior part is arranged around the testicular artery. The middle part lies around the ductus deferens and its artery, and the posterior part is isolated. This plexus condenses into four veins at the superficial inguinal ring and into the two veins at deep inguinal ring. These veins run along testicular artery, and ultimately one vein is formed which drains into the inferior vena cava on the right side and left renal vein on the left side. So here the left on the left side drains into the left renal vein. The varicocele is produced by dilatation of the pemphiform plexus. It, it is most common on the left side, possibly because of the left testicular vein is longer than the right and enters the left renal vein at right angle and is crossed by colon, which may compress it when it is loaded with the fecal matter. Here in this diagram you can see the blood supply of the testis. This this is the you can see is the testicular artery early coiled, and this divides into medial and lateral branches. Here you can see it is present with the appears that uh, tunica albuginea, and they run on the sides and form the tunica vasculosa. They, here you can see the testicular artery is surrounded by the pemphiform plexus. This is the pemphiform plexus of vein, and here with what you can see the artery of the vas deferens, and this is the cremastic artery. This color Doppler study has been done to see the pemphiform plexus, and here what you can see the varicocele, they are dilated pemphiform plexus. Lymphatic drainage. The lymphatics from the testis ascend along the testicular vessels and drain into the pre-aortic and para-aortic lymph nodes at the labella of the second lumbar vertebra. Nerve supply of the testis. The testis is supplied by sympathetic nerves arising from T10 through renal and aortic plexuses. These nerves, they are afferent for the testicular sensations and they are efferent for blood vessels, and they are vasomotor in nature. Development. The, the gonadal ridge is formed by proliferation of the epithelium and mesenchyme. The primordial germ cells originate in epiblast and reach the genital ridge through the dorsal mesentery. The epithelial cells penetrate the underlying mesenchyme to form the primitive sex cords before arrival of the primordial germ cells. The SRY gene influences the primitive sex cords to proliferate into deeper aspect to form the medullary cords of the testis. At the hilum, the cords break up to form the retitestis. The fibrous connective tissue, that is a tunica albuginea, separates the cords from the surface epithelium. The sustentaculum cells of the lady are derived from the mesenchyme of the gonadal ridge. After puberty, lumen develops in the cards, and these cards are transformed into the seminal vestibules. The seminal vestibules join the retitestis. The mesonephric or the Wolffian duct, after linking with the retitestis, forms the ductus deferens. Here in this diagram, you can see the formation of the genital trees at 6 weeks. Here you can see there is proliferation of the surface epithelium, the coelomic epithelium, and the sex cards they are formed, and they proliferate and migrate and protrude into the uh, surrounding the present mesenchyme. This is what you can see is the Wolffian duct. This is the mesonephric tubules. Now, at about 8 weeks, you can see the mesonephric tubules they degenerate, the Wolffian duct persists, the cards, the sex cards, they are the testicular cards, they proliferate further into the deep into the mesenchyme, surrounding mesenchyme. At 20 weeks here, you can see the tunica albuginea that is found by the surrounding mesenchyme separates the testicular cards from the surface epithelium, and here you can see the posteriorly the cards they anastomose with each other, forming the retitestis, and this retitestis communicates with the Wolffian duct by the efferent ductus. Here you can see the efferent ductus. Here you can see at 16 weeks the there is formation of tunica albuginea separating the testis cards from the surface epithelium. Here at the posterior border, these testicular cards they anastomose with each other forming the retitestis, and this retitestis is connected to the Wolffian duct by the efferent ductus. So these are the efferent ductus. Descent of the testis. The urogenital mesentery attaches the testis to the posterior abdominal wall. The caudal part forms the caudal genital ligament and gubernaculum. When testis reaches deep inguinal ring, gubernaculum extends from the inguinal region to the scrotal swelling, and finally it contacts the scrotal floor. The factors controlling the descent of the testis are the outgrowth of the gubernaculum, number 2, increased intra-abdominal pressure, number 3, regression of gubernaculum, number 4, androgens and Mullerian inhibiting substance, then evagination of the peritoneum as processus vaginalis into the scrotal swelling results in the formation of the inguinal canal. The testis reaches the inguinal region at about 12 weeks, the inguinal canal at about 28 weeks, and scrotum at about 33 weeks. Coverings of the testis are derived as such as the processus vaginalis gets attached from the peritoneum, gets detached from the peritoneum and forms the parietal layer of tunica vaginalis. The transversalis fascia forms internal spermatic fascia. The internal oblique abdominis forms the cremastic fascia in muscle, and the external oblique abdominis form the external spermatic fascia. Here you can see the clock for the descent of testis. At seventh week, you see the caudal genital ligament here. This is caudal genital ligament. This is the testis, and this is what you can see is the gubernaculum, and this is here is the scrotal swelling. At 12 weeks, here you can see the formation of the vaginal process from this peritoneum. At 9 months, you see the testis is present in the scrotum. Histology of the testis. The seminiferous tubules consist of cells arranged in 4 to 8 layers in fully functioning testis. These cells are of two types. Number one is spermatogenic, and number two supporting are the sustentaculum cells of Sertoli. The spermatogenic cells have vast majority and include spermatogonia, the primary spermatocyte, the secondary spermatocyte, spermatids, and spermatozoa. The cells of Sertoli are tall columnar cells extending from the basal lamina to the central lumen. They support and protect the germ cells and help in maturation of spermatozoa. The interstitial cells are the cells of Leydig. They are found as small clusters between the seminiferous tubules. They secrete the testosterone. Here, this is the H&E stain section of the testis. Here you can see the cavity of the tunica vaginalis. This is the tunica albuginea covering the testis, and these are the seminiferous tubules. You can see the lumen here. Here is the lumen of the testis and seminiferous tubules. Here, the here in the seminiferous tubules you can see the cells. The lining cells are arranged in 4 to 8 layers. These are the germ cells consisting of spermatocyte, primary spermatocyte, secondary spermatocyte, the spermatogonia, the spermatids, and the spermatozoa. And these tall cells you see here in between, these are the sustentacular cells, and here what you can see is the interstitial cells of Leydig which secrete the testosterone. These are the clusters of cells present in between the seminiferous tubules. Epididymis. Epididymis is an organ made of highly coiled tubule that acts as a reservoir of the spermatozoa. It is said to have a head, body, and tail. The head is enlarged upper end connected to the upper pole of the testis by efferent ductus. It is made up of highly coiled efferent ductus. The body of the testis is the middle part and tail is the lower part. The body and tail are made of a single duct, the duct of the epidermis, and this duct of the epidermis is highly coiled on itself. At the lower end of the tail, this duct of the epidermis becomes continuous with the ductus deferens. Here in this dissection, you can see the testis, the epididymis here. This is the upper end, this is the head, this is the body, and this is the tail of the epididymis. And this tail is continuous with the vasa deferens or the ductus deferens. This is the artery of the testis. Vessels and nerves. The epididymis is supplied by testicular artery through a branch which anastomoses and it reinforces the tiny artery to the ductus deferens. The venous and the lymphatic drainage are similar to those of the testis. Then, similar to the testis, epididymis is also supplied by sympathetic nerves through the testicular plexus containing T11 to L1 sympathetic nerves. Histology of epididymis. The epididymis consists of ductules. They are lined by pseudo-stratified columnar epithelium with the stereocilia, and they are having stereocilia forming the breast border. The tubules are surrounded by connective tissue. This is the H and E stained section of epididymis. You can see here the duct of the epididymis. This is the lumen. In between the duct of the epididymis, we find the connective tissue. Then this duct of epididymis is lined by pseudo-stratified ciliated columnar epithelium. Here you can see the stereocilia forming the breast border. Ductus deferens. Ductus deferens is a thick-walled muscular tube extending from tail of the epididymis to prostatic urethra. Distal part of the ductus deferens is dilated to form ampulla, which joins the seminal vesicle to form the ejaculatory duct. The right and the left ejaculatory ducts pierce prostate and open into the prostatic urethra. This ductus deferens is developed from the mesonephric duct. Outside the superficial inguinal ring, this ductus deferens can be palpated in the scrotal sac. Here it can be cut and ligated. And in the operation of vasectomy. So here in the operation of vasectomy, the vas deferens is palpated in the scrotal sac, cut and ligated. And this vasectomy is the permanent method of the family planning. Histology of the ductus deferens. The wall of the ductus deferens is composed of three coats: the inner mucosa, middle muscular layer, and the outer adventitia. The mucosa, it comprises of pseudo-stratified columnar epithelial cell lining and underlying lamina propria. The epithelium is provided with the stereocilia. The mucosa is thrown into longitudinal folds which permit expansion of the duct during ejaculation. The muscular layer, it consists of smooth muscle fibers. They are arranged into an three layers: the inner longitudinal, middle circular, and outer longitudinal. The adventitia, it is made up of fibroelastic connective tissue carrying blood vessels and the nerves. Here you can see the HNE stain section of the vas deferens. This is the lumen of the vas deferens, and you can appreciate it is a thick walled muscular tube with a small lumen. Mucosa here you can see shows the longitudinal folds. This is lined by the pseudostratified ciliated columnar epithelium here, and this has got the stereocilia. This is the what you can see is the lamina propria. This is what you can see is the lamina propria. This is the muscle layer. Here is the inner longitudinal, middle circular, and this is the outer longitudinal. So this is all about the male external genitalia.